Good morning, everyone. Welcome to the Unit 2 review. For this review, we're going to be covering Chapter 4, which is Biopsychology, Chapter 5, which is the Stress, Lifestyle, and Health chapter, and Chapter 6, which is all about learning theory. All right, so let's get started. And I'm going to move really quickly because I know you want to keep these short, right? All right, so here we go. So this slide right here is demonstrating the neuron. This is the basic brain cell. It's estimated that we have about 100 billion brain cells in our brain. And I have asked that you learn at least the 4 basic parts, and then we'll talk a little bit about the 5th part, which is called the myelin sheath. The beginning of communication with the cell starts with the dendrites. So let's go ahead and start over here with the dendrites. So all these little extensions over here that you see that look like little roots of a tree, those are called dendrites. And dendrites have a primary function, which is to receive incoming information. And that information is represented in the form of a neurotransmitter. So these dendrites are constantly receiving neurotransmitters. And all right, so dendrites receive, and then the next step for neural communication is the cell body, also called the soma. And what you can see here, it is the part of the cell that contains the nucleus. So this cell body is really important to keep it alive. Anything that will attack it will kill off the neuron for good. Whereas dendrites or axons can be damaged, and because of my favorite word called neuroplasticity, they can recover and they can come back. But once the cell body has been attacked, that neuron is gone forever. And things that attack the cell body, I have to say anything that cuts oxygen supply to your brain cells, anything that reduces the oxygen supply in your bloodstream, anything like blockage for an artery, a stroke, any of those things can lead to cell death of the cell body. And so we want to be careful with all those things, right? And of course, a head injury kills off a lot of cells as well. All right, so from the base of the cell body that you see here, extends this long, thin fiber called the axon. And the axon is where conduction takes place. And we're going to be looking at that one more specifically in a few slides. So the axon is the long, thin fiber that extends from the cell body and ends in the axon terminal buttons, these little alien-like fingers that you have down here on the bottom. And these terminal buttons are important because this is where our neurotransmitters are stored. So we will be looking at those in a closer way as well. Finally, just to cover the myelin sheath, the myelin sheath is this fatty tissue that protects the axon. Not all of our neurons are covered in myelin sheaths. The ones that are myelinated tend to be primarily our motor neurons. And so one of the things that the myelin sheath does is it speeds up neural transmission. So it makes these neurons faster acting. And that's very common for our motor neurons, especially as we're learning something new. If we're learning like a new dance step or how to play a musical instrument, our neurons will myelinate. And as they become myelinated, we will get faster and smoother at doing those processes. So my favorite word, once again, just so you know it, is neuroplasticity. And this means that neurons can adapt, they can change, they can reorganize and restructure themselves. Those dendrites from one neuron could be in the thousands. And so if you need that neuron to do something, it is able to do it. And likewise, if you stop asking that neuron to do something and you're not really challenging your neurons, they retract as well. So we want to make sure we're keeping our neurons nice and challenged every day, which is great that you're taking this class, right? All right, so when it comes to neural communication, most of the time people talk about electrical processes. And granted, they are electrical in one respect, and that is that we're going to have some charges that are involved depending on the inside and the outside of the neuron. But the actual cause of these electrical charges is based in chemicals. And so we're going to be looking at those closely. So this is what it means when a neuron is at rest. It actually has a charge on the inside of the axon of a negative 70 millivolts. And these millivolts, at the negative 70, it is said to be at rest. When a neuron is at rest, it is not activated. So it is constantly receiving incoming messages because, as we mentioned in the previous slide, it has thousands of dendrites and they're constantly receiving incoming information. Neurotransmitters can communicate in one of two ways. They are either excitatory, which means they make the receiving neuron want to have an action potential, or they are inhibitory, and it makes the receiving neuron stay at a resting potential. If enough excitatory neurotransmitters are received by a neuron, then it will increase the charge, and anything that causes it to go over this negative 55 stimulus threshold is going to lead to what we call the action potential. And the action potential operates on what is called the all or none principle, which means once it crosses this threshold, it's going to have an action potential. And this actually involves the opening of the sodium ionic channels. And so we'll be looking at that in a few slides. Once it reaches its full action potential, which is a positive 30 millivolts, according to your book, then it will begin to repolarize. And what that means is we're going to need to get everybody back in position. And one of the effects of putting everyone back in position is this overshoot effect that you see here at the bottom. And so a very brief period of time, the neuron is actually even lower in charge. It's projected to be at a negative 90 millivolts. And during that time, it is said to be hyperpolarized, and it cannot fire during hyperpolarization. So the neuron must reestablish a resting potential so that it can be ready to fire again. Let's look at that a little more closely now, as far as the chemicals that are involved. And there's two primary chemicals that are involved. There's more, but there's two. We're simplifying things. And so the two primary chemicals that are involved in this process are sodium and potassium. Sodium is typically living on the outside in the extracellular fluid, and potassium is typically living on the inside. And so if you're my students that have me for my face-to-face class, you know that I talked about this kind of being you on the inside. You were potassium and you're at home and you're receiving all these incoming calls that are saying excitatory, let's have a party, or they're inhibitory, let's not have a party. But when you receive just enough and more excitatory than inhibitory neurotransmitters, these little red lines here are representing ionic channels. In class, I call them gates. And so once it crosses that stimulus threshold, then what's going to happen is you basically are going to open the gates in a domino-like fashion. And this would be the axon, the cell body. We can imagine it over here on the left would be the cell body. And it's going to travel down in a domino-like fashion, and it's going to end in the terminal buttons. So let's see what happens during an action potential. So I call it the big party because you're getting those phone calls. Hey, let's have a party. Let's have a party. And if you cross the threshold at the negative 50, negative 55 millivolts, then that means you're going to open the gates. And guess who gets to come inside? So now there you were at home, comfortable, potassium, and now suddenly sodium gets to join you on the inside. And typically sodium's on the outside. And so because they're both positively charged ions, now when they get together, they dramatically change the inside charge of the neuron. And so it brings it up from a negative 70 during the resting potential to a positive 30 millivolts during the action potential. And the main thing you want to remember about this is as sodium comes in and joins potassium and they have this big party, the whole purpose of this is so that at the end of this axon over here on the right are those little terminal buttons, and the end product of the action potential is the release of the neurotransmitters. So it basically has this whole party and lets in sodium so that it can release the neurotransmitters, and then it quickly ends. The party comes to an end. All good things must come to an end, right? And so just as quickly as the party happened, everyone gets kicked out. And I mean, I'm oversimplifying it. If you ever decide to become a neuroscientist, you're going to see that this is actually a more complicated process. But in the end, you do have potassium outside for a brief period of time, along with sodium, who tends to leave a little more passively. And this drops the charge, and this is what causes the hyperpolarization. So those are the three phases of neural activation. During this final period, this recovery period down here at the bottom, the neuron cannot fire. It is not ready to have another action potential. Why? Well, because if you want to picture potassium as the owner of the home where the party is taking place, you got to have potassium on the inside so that potassium can open the gates, right? And so maybe that'll help you remember. We got to have everyone back in position, and then it starts all over again. And by the way, this process doesn't take very long. We're talking a neuron is capable of firing up to 1,000 times per second, right? So it is a very, very fast process. All right, let's keep going. All right, so once those neurotransmitters are released, they're released into what you see here as the gap. So this is a picture of the terminal button and the release of the neurotransmitters. So neurotransmitters are stored in these little bubbles that you see here called synaptic vesicles. And once they are released into this gap, this cleft, what's called the synaptic cleft, it is basically a meeting place for this terminal button to meet up with this dendrite from another neuron. And so here you want to notice that the synaptic vesicles kind of rise to the surface and they get released during the action potential. They get released into the synaptic cleft, and depending on their purpose and their function, they either bind to the receptors on the dendrites, and either are they an excitatory or an inhibitory neurotransmitter. So that's how they work. You guys give me 1 second. I'm sorry, my dog is crying and I think I need to let him out. All right, so here we go, keep going. And the last thing that has to happen is reuptake. And so once those neurotransmitters are released into the synaptic cleft and they either trigger, bind, or block, they can have different functions on the receiving dendrites. Whatever is left in the cleft is taken back up through a process called reuptake. And so these little bubbles, if you can picture these synaptic vesicles kind of starting off in the morning after you've had a full night's rest, they're nice and full of neurotransmitters. And so as you function throughout the day and these fire multiple times per second constantly throughout the entire day, you can start picturing that those levels begin to run low. And so the lower the neurotransmitter count that you have, the more likely you are to feel irritable or to start making mistakes and not be at your best mentally, emotionally, even physically, because neurotransmitters have all of those functions. And so, yeah, that's why it's so important that we sleep and that we sleep enough. Also, we don't want to oversleep. So like maybe sometimes you've kind of just been really exhausted and you sleep and you end up sleeping like 12 hours. You don't usually wake up feeling refreshed after something like that. If anything, you might wake up and you feel tired and groggy and like this fog has come over you. And that's because you overstocked on these neurotransmitters. So I call it the Goldilocks effect, that you don't want to have too much and you don't want to have too little, right? We need just the right amount of neurotransmitters. These are some videos that are linked through your book that I wanted to recommend for you. One of them is called Lights, Camera, Action Potentials, and the other one is called The Chemical Mind. So please make sure you watch them. They are linked in your book. Okay, I'm not going to take up time on this video showing them, but just make sure you come back and watch them. So, all right, so we already mentioned the synapse, and the synapse is that meeting place, right, where the terminal button comes to meet the dendrite of another neuron. We already mentioned what neurotransmitters are, that they are the chemical messengers that either trigger an excitatory process on the receiving neuron, or they are inhibitory and they keep it from firing. And then finally, these are like, picture these green little buttons over here are representing the receptors. And so the receptors are these little proteins as well, where the neurotransmitters attach, and they have their differing functions. Now, these neurotransmitters are many. We have over 100 that have been discovered in the brain so far. Some of my favorites, of course, my number one is dopamine, and that one is most involved in pleasure and learning and mood. GABA aminobutyric acid, is the primary inhibitory neurotransmitter. It is most prevalent, the most prevalent inhibitory neurotransmitter in the brain. And it is linked as well with alcohol. So alcohol leads to the release of GABA, and this is one of the reasons why you can end up with alcohol poisoning. Glutamate is the most prevalent excitatory neurotransmitter in the brain. And many of you might have met Sarah, good old Sarah, serotonin. And serotonin is linked with mood and mood disorders. So when we study like depression, you're going to hear about like serotonin, selective serotonin reuptake inhibitors, or what are called SSRIs. And those are one form of antidepressant drugs. So anyway, you want to know the major neurotransmitters here on this chart. They were given to you in your book, and then just kind of be familiar with some of their effects. As far as organization of the nervous system, we have the central nervous system, which includes the brain and the spinal cord, and the peripheral nervous system, which includes everything on the periphery, right? That is not the brain and the spinal cord. That gets further subdivided into, from the peripheral, we see a division into the autonomic versus the somatic nervous system. The autonomic, you want to think of all the internal, visceral, organ, gland things that get done for you automatically. The somatic nervous system involves control of the muscles and skin. And so somatic tends to be much more voluntary. Autonomic tends to be much more involuntary functions. Autonomic finally is subdivided into further subdivided into two sub areas. So we can take the autonomic and we see the sympathetic nervous system that is part of it, and the parasympathetic. So just keeping in mind that when it comes to the sympathetic nervous system, we are looking at things like fight or flight. And when we're looking at the parasympathetic, we are looking at our normal functioning. So keep in mind the organization of the peripheral nervous system, right? Okay, so somatic versus autonomic. Autonomic gets subdivided into the sympathetic and the parasympathetic, and you want to know what those do. So when we are calm and at peace, as was described in class, then we have basic digestive processes are happening. If you had a nice lunch, you're processing your lunch, your stomach is, munching it out and sending it off to your intestines. Your breathing is normal, your heart rate is normal, your bladder is normal, you're able to control those things. So that's what you would look like during you at peace, right? If you can picture you during normal functioning, that should be your normal. If, however, you see a bear and you're out hiking and this bear wants to eat you, then it would trigger the sympathetic nervous system. And the sympathetic nervous system begins with pupil dilation, increased heart rate, because we're trying to get all this blood running to your appendages, like your legs and your arms, so that you can fight or flight. And so if you are needing to fight, you are going to need increased oxygen. So you start breathing faster. Digestion is not important at this point, and so the blood is redirected from your stomach over to your legs and your arms. And this is why sometimes when you are having some stress or you're nervous, maybe you're going to give a speech in class or something, you will feel butterflies in your stomach, and that's because blood is going, instead of from your stomach, it's getting redirected over to these appendages. And then, of course, you get dry mouth, cotton mouth sometimes when you're nervous, and that has to do with sympathetic arousal as well. And good thing, I mean, I taught speech, and nobody ever, like, you know, peed themselves in a speech, but it is possible, right? Depending on how scared you are, right? And so, okay, so those are the things that the sympathetic nervous system does for you. Keep in mind, it's trying to help you survive, right? So, all right. The next part of the chapter goes into the endocrine system, and the endocrine system is ruled and controlled by the hypothalamus. The hypothalamus is in the brain. We're going to see it in a brain tour that I'm going to give you right now, but it is considered the primary liaison between the brain and the endocrine system. And endocrine is mainly your hormonal system. So what you're seeing here as far as these organs that are being pointed out to you, such as the thymus, the pineal gland, the adrenal glands, the pancreas, the uterus, the ovaries, the testes, these are all differing organs that produce different kinds of hormones. And so your endocrine system has to do with your hormones. And there is a slide coming up, I guess, on the hormones. So I will show you that one in a second. I thought it was right there, but we'll jump to it in a second. Your brain, as far as your brain is lateralized, which means you have a right hemisphere and a left hemisphere. Kind of get the biggest, broadest organization of the brain, then you would start here with having 3 parts to the brain: forebrain, midbrain, and hindbrain. Those are the biggest ways to organize the brain. But of course, there's a lot of subparts that go within there. So we're going to get a lot more specific than that. Right, so looking at the hindbrain actually includes 3 important structures. The medulla, which does all those really important autonomic things for us, such as heart rate, breathing, blood pressure. The medulla basically is keeping you alive without you having to think about it. So that medulla is really important, right? Next to the medulla is the bulging part right here called the pons. And the pons is connecting, first of all, your brain and your spinal cord. It also has striations that reach out to the cerebellum. And then your cerebellum is your center for balance, coordination, motor memory. Your ability to dance a certain dance step is here in your cerebellum, right? Okay, and so once your cerebellum is affected, if you're drinking alcohol, you will be in a stupor and you will not be able to stand upright. So that's pretty dangerous, because if that's how drunk you are, think of how close you are to the medulla. So once again, speaking of alcohol poisoning, and when you have someone that's no longer able to stand up, is in a stupor, or even worse, passed out, then that means their medulla is in danger and they are in danger of death. And that's when you should definitely be calling 911. right? Okay, or driving them to the hospital if they're in your presence. Okay, the other parts, the other parts of the three subdivision was the midbrain. In the midbrain, we have two areas that are important. One of them is called the substantia nigra, and this is a very darkly pigmented area of the brain. And it's darkly pigmented because it stores dopamine. So this is an area where if you went into this section, no matter day or night, you would find plenty of dopamine. And dopamine involves our motor as well as pleasure and our reward system. So this is a primary neurotransmitter. As for the reticular formation, that one has to do with our sleep-wake, our alertness, how aroused we are as far as being awake and alert. And so that reticular formation is important, and it is said to play a role in attention deficit disorder. And so being able to recognize what you should pay attention to, what matters, what regulated, what information should be regulated is part of the role of the reticular formation. And this is why we give, well, we don't, psychologists don't, but psychiatrists tend to give kids who have ADD and ADHD, they tend to give them stimulants such as Ritalin or Dexedrine or Adderall. And that's because it tends to trigger the reticular formation and activate the reticular formation so that it can do its job. All right, so the outer crust of the brain is called the cortex, and the cerebral cortex is divided into 4 lobes. We know that the frontal lobe is the seat of logic, reasoning, planning, personality is said to be stored in the frontal lobe, and some motor functions, and just have this primary big fold that you see here right past the central sulcus, and that is the motor, the area of motor, the primary motor strip. On the back of the central sulcus, this big fold that you see here is the parietal lobe, and the parietal lobe is said to be directly linked to our sensory ability with our skin and sensation of touch and temperature and pressure. So that is our parietal lobe. Back here in the back, we have our occipital lobe, which has to do with vision and recognizing visual patterns. And then what you see here in green is the temporal lobe, which would be closest to your temples if you're envisioning this brain inside your head. And the temporal lobe, since it's closest to your temples, maybe you can make the connection that it has to do with auditory systems and as well as language and speech. On the frontal lobe, I was speaking to you about the central fold called the central sulcus, and towards the front of it is that primary motor strip, right? And so that motor cortex plays a role in coordinating movement. The prefrontal cortex is where we see like the really higher order cognitive functioning taking place. So the real, you know, the creativity, the logic and the problem solving, those are all happening in the prefrontal cortex. And then they are pointing out Broca's area, but this picture does not show it to you. But Broca's area is part of frontal lobe that has to do with language production. And sometimes people have a stroke that affects their Broca's area, and therefore they will have speech production issues. So they may slur their words, they may not be able to pronounce any words afterwards. So you will see that something is happening with their speech production. As for the parietal lobe, we mentioned that it is a primary somatosensory area, and it literally has to do, it is organized what is called topographically, which means that there are actual spatial relationships that exist through labeled lines that exist through the body and are maintained on the surface of this somatosensory cortex. The temporal lobe we mentioned is part of hearing, auditory processes, and it also holds something called Wernicke's area. And Wernicke is involved in the comprehension of speech, whereas Broca's area is involved in the production of speech. And finally, the occipital lobe is the primary visual cortex, and that is where we will see issues like show up possibly with our visual field, being able to recognize that an object is what it is, right, through our representation in our cortex. All right, now let's go a little deeper, a little further below the surface. So that cortex is on the surface. And so that is, when we talk below the surface, now we start talking about subcortical structures. And one of the big important ones that we want to know that is considered part of the forebrain is the thalamus. So the thalamus here in this picture is represented by this big yellow structure that you see here. And it is considered the primary relay station for all sensory information except smell. Okay, so everything except the sense of smell is routed up the spinal cord, through your thalamus, and out to the corresponding areas of your cortex. And then again, your cortex decides, brings it back to the thalamus, and sends it out to your whatever sensory or motor systems that you are working on. That's the thalamus. Okay, and then the other parts of the limbic system, in addition to the thalamus, include the hypothalamus. But hypo means under, so the hypothalamus is situated below the thalamus. The amygdala, right, okay, which is considered like an almond-like structure at the base of the hippocampus. And the hippocampus is this long, they sometimes call it seahorse-like structure. And so the hippocampus, very important when it comes to memory, right, especially episodic memories and new memory formation. The amygdala has to do with emotion and very specifically negative emotions, such as anger, jealousy, fear, right? And then the hypothalamus, and this area right here below the thalamus, not very large, much smaller than the thalamus, but it is a primary player in many of our biological processes and homeostatic processes, such as hunger, thirst, sexual motivation. It even plays a role in sleep. And we mentioned earlier that it is what is linking the endocrine system to the brain. And then this is just a picture that might tie it all up together for you. So we're seeing here, if we go from the hindbrain structures, there's the medulla, there's the pons, and there's the cerebellum, all considered hindbrain structures. Here's the occipital lobe. When we come to the cortex, there's the parietal lobe. Here's the big central fold down the hemispheres, and it is called the central sulcus. To the front of it is known as the precentral gyrus, or that primary motor strip. To the back of it is the postcentral gyrus, or what is the somatosensory strip. Here is the frontal lobe, and the underlying structures, the corpus callosum, the thalamus, the hypothalamus, and any other structures that I think might have been pointed out to you in your book. Maybe they mentioned to you, I don't think they did, but maybe they mentioned the optic chiasm. They will when you look at the vision system, but yeah, okay, so that's a great way to wrap it all up there. As far as imaging the brain, we have various ways, and it kind of depends on what we're trying to get to, but we do have a CT scan that we can do. We have an EEG, an electroencephalograph. We have fMRIs, which are functional magnetic resonance imaging that can be done while you are thinking. They can show like your brain in action kind of thing. Of course, they're very expensive, but still. We also have MRIs, and we have PET scans. And it kind of all depends on what the doctor or whoever is requesting the scan is trying to see or address, right? Okay, with your brain. But yeah, those are all different ways that the brain can be assessed. All right, so this picture here, I'm going to give you just a few seconds here. You can look at it and test yourself. Use it to test yourself to see if you understand the outer brain structures of the cortex. All right, hopefully, let's see. All right, so I would hope this one was pretty easy. Frontal over here, right? Parietal over here on the top right. In the back over here, occipital over here on the bottom left, temporal. here we have the brainstem. It is specifically pointing to the medulla, and there's no arrow there, but there should have been maybe one to the pons. And then this little mini brain bulge back here is the cerebellum. So let's see, you got it, right? Okay, so hopefully you got all those right. Here's another way to test yourself. So we have the outer cortex, which is great. We just saw that on the previous slide. And then now if we go below subcortically, we start talking about like the big bulge in the middle would be the thalamus, right? Below the thalamus is the hypothalamus, right? Okay, we have over here we have the cerebellum, we have the corpus callosum, right? Here we have the pons and the medulla, and this arrow right here is actually pointing to something called the midbrain. And then this little structure right here, number 5, is the hippocampus, right? So let's see, did we get them all right? I hope so, right? Okay, so yeah, test yourself with these, right? And there's plenty more online if you want to play with those, but it's always great to color them. It's always great to kind of test yourself so that you can check and see if you learned what you were supposed to learn. All right, some other parts to the chapter, it kind of goes into a bio section. Give me a second. And it goes into some conversation on genetics, and it didn't give you very much, but it's just kind of talking about the word epigenetics and how, you know, the genetics are kind of like the foundation that we're all built upon, but they constantly interact with our environment. So we could have the gene code for being really, really tall, but if we don't have any milk available and calcium, then we're not going to fulfill our genetic potential, right? So those are important. Okay, we're done with the biopsychology chapter. Yay. Okay, moving on